This short clip demonstrates how FGF2 binds to FGFR on the cell surface. FGF2 binding is stabilized by heparin sulfate proteoglycans, and receptor dimerization leads to phosphorylation of the receptor tails, activating two major signaling pathways, the MAPK/ERK pathway, which drives proliferation and differentiation, and the PI3K/ACT pathway, which supports cell survival and migration. FGF2 directly stimulates endothelial cell proliferation and migration, inducing angiogenic sprouting. At the same time, it upregulates VEGF and other proangiogenic factors, bringing blood, oxygen, and nutrients to the repair site. FGF2 acts as a chemotactic factor, guiding fibroblasts, mesenchymal stem cells, and vascular progenitor cells to migrate toward the injured area and activating them into a repair state. FGF2 strongly stimulates fibroblast proliferation and promotes the synthesis of collagen and fibronectin, thereby constructing the three-dimensional scaffold of granulation tissue, which provides the structural foundation for new tissue. FGF2 accelerates epidermal cell proliferation and migration, promoting reepithelialization. In bone tissue, it promotes osteoprogenitor cell proliferation and matrix mineralization. In the nervous system, it provides trophic support and improves the repair environment. In the early stages of repair, FGF2 modulates macrophage function, promoting partial conversion from M1 to M2 phenotype, establishing a microenvironment favorable for healing. Clinically, FGF2 is applied in the form of recombinant topical gels or sprays for chronic non-healing wounds, where it directly stimulates granulation tissue growth and epithelial regeneration, accelerating wound closure. Cutting-edge research is exploring the combination of FGF2 with hydrogels, microspheres, or gene therapy for localized delivery to ischemic myocardium, nerve injury sites, or tissue engineering scaffolds, thereby promoting angiogenesis, axonal regeneration, and functional repair.